Hi, everyone, and welcome. My name is Brooke Christensen, and I'm a PhD student in the Daily Lab in the Department of Ecology and Evolutionary Biology at the University of California, Irvine. I'm happy to welcome you to the third in a series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the National Science Foundation Division of Organismal Systems, with the goal of bringing together scientists across disciplines at the intersection of organismal biology and physics. We aim to highlight the use of model-based and mathematical tools to address fundamental questions in organismal biology. The workshops will be recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences with the School of Biological Sciences. And with that, please welcome today's workshop speakers, Dr. Laura Miller from the University of Arizona and Dr. David Hu from Georgia Tech. Thank you very much.
So this is our first workshop focusing on fluid organism interactions, and we are going to talk about mathematical and numerical approaches to these problems. So everything from dimensionless groups to computational fluid dynamics. And in 2 weeks, our second workshop is on measuring flow experimentally. So please come to that. It's Friday, April 29th from 10 to noon. And Arvind Santhanarakrishnan, Eric Teitel, Saad Balmla, and Matt McHenry will be presenting. And I know that they will be taking you into the lab to look at processes for PIV and then showing you how to analyze the velocity vector field. Okay, so just to introduce the mathematical and numerical approaches and to tell you a little bit about the benefits of simple models as well as computational fluid dynamics, I want to introduce just in general the philosophy of mathematical modeling or the art of mathematical modeling. And there is this great quote from George E.P. Box that all models are wrong. And the practical question is, how wrong do they have to be not be useful? Or more simply, all models are wrong, but some are useful. And this is an idea that in education, we don't really drive home so well. If you look at the general process of mathematical modeling, you can imagine that you have the investigation of a problem and its identification. So let's say that you want to understand how a fish swims. At this point, if you're going to make a model, you need to put in some assumptions and selections of inputs and outputs. And there are always assumptions to be made. So you can't possibly have all the complexity of a fish or another organism in your model. And what are you going to put into the model and what will be the output? And then from there, you could mathematically formulate that problem, solve your math model, interpret the solution, compare with reality, maybe refine your assumptions, and eventually write and present the results. In our curriculum, we mostly just focus on this bottom part, where you have the mathematical formulation of a problem, you solve it, and you interpret the solution. And another limitation, just in terms of teaching large classes, is that you're given mathematical formulations that can be solved either analytically or they're computable or they're nice. And it's easy to get the impression that everything has a solution, everything can be computed. And this is actually pretty far from the truth. Most things cannot be solved analytically, or we don't know how to solve them. And it's really easy to make problems so complicated that they're incomputable. So you end up having to make a lot of choices in order to actually be able to say something about the system that you're looking at. And so to start, we're going to look at some simple models and dimensionless groups. And just to point out Steve Vogel as one of the grandfather of this field in terms of simple modeling in fluid organism interactions. And I will make these slides available because I have quite a few links to books and software, but he was very prolific during his life, writing a number of books for the community. And so from here, I will hand things over to David.
All right, thanks a lot. It's a great pleasure to share this workshop with Laura. Laura and I go back many, many, many years, back to Courant Institute, and I think we're just kind of have complementary approaches. She'll talk about real sort of, I was going to say hardcore that would make what I'm doing softcore. But she's going to do CFD, and I'm going to do scaling. So computational fluid dynamics is really looking at the detailed flow patterns. I'm going to teach you a few ways you can figure out physical principles by sort of looking at just the basic length scales and time scales involved. In some ways, scaling, this process has been around for a long time. Capillarity, this topic of looking at fluid surfaces and interaction with solids, that was made popular by the Nobel Prize winner, de Gennes. And if you want to read more, he has a book with two other co-authors, Francois Bouchard and David Craig, called Capillary and Wetting Phenomenon. And he says explicitly, I'm going to have very little math in this book. So if you want to learn about the big picture of the capillarity, you can check that book out. Today, I'm going to give a couple case studies in which we basically start with knowing very little about a system and going to looking at the length scales and the time scales involved to basically getting an understanding of how the system works. And this is the work done by my graduate student, Andrew Dickerson, who's now at Tennessee, and Alexis Noel, who's at GTRI. So my research methodology for the projects I'm going to talk about basically involves finding a model organism. Some people know that as Krog's rule, where you look at a champion animal. And that organism will really dictate the ease at which you'll be able to do these experiments and the ease at which you'll be able to extrapolate results to other animals in the order. You want to find an unsolved problem. So hopefully this organism performs a task that is difficult to observe with other animals, but for this organism, it's small enough or slow enough that you can actually observe it. The next step is we usually do just a few simple observations. A lot of people think modelers work sort of devoid from the experiments, but I always heard that don't try to model something unless you have an idea what the answer should be. And you get an idea of the physical intuition of the problem based by just doing a few simple observations. So something that hopefully is cheap and easy and that you can get an answer quite quickly so you can inform your modeling. So I'll show you a few tools that I use to do what's called a scaling analysis. So basically prediction of the sizes and the prediction of the speeds of these animals as a function of body size, and how they can help us develop these quantitative hypotheses, or ways that we can predict, do these things get faster or slower as they get bigger or smaller? And then the rest is a process of iteration. A lot of times we do experiments with mimics. I'll show you, we built this wet dog simulator, this device that can sort of shake hair on a dog and release water, and how experiments with often these mimics can give us a sort of a more complete picture and confirm what we saw with the animals. Nice thing in this field is that there's a lot of data on different animal sizes in museums and literature, and I'll show you how to combine that with mathematical modeling to basically, again, support your physical picture. And then through this process of basically going back and forth between the experiments, the museum specimens, and for me, your simple, hopefully simple and pretty straightforward model, you can get a simple, coherent physical picture. So something that makes sense across all these different topics. And Laura will show how if you can do CFD to also get even more detail, and also something that also gives you. And in my view, if you do this right, the process takes about a year, going back and forth doing experiments and modeling. And that's partly because my models are quite simple compared to Laura. So I've used the word scaling. I'm not talking about scaling your bathtub or descaling a fish. I'm talking about how things change with orders of magnitude and sizes. For example, so scaling is basically an interdependence of physical parameters. Physical parameters are things you can measure. For example, on the right here, we have a work from Sullivan's paper on bird flight, where you see the wing loading, which is the weight per surface area on the wing, changes with body weight. So this is well known that you can build these plots that go from birds to airplanes to insects. And that's the beauty and the power of scaling analysis, that it can give you insight across a huge number of animals and even artificial systems. Laura and I were talking that the best books on this subject are actually out of print because we're so old now. So 1984, Scaling, this is a great book. It's out of print. On Size and Life, these are two books that you should have on your shelf if you're interested in this process. So the fundamental idea of why scaling, and as I'll introduce dimensional analysis, works is that physical principles appear across body size. There's this idea of dynamic similarity that for you can have two almost, to be honest, I teach this to my undergrad students. I still see that as magic. On the left here, you see an aerial photograph from a satellite of Guadalupe Island, and there's a mountain range on the island that actually disrupts the incoming wind. And what you see in the wake is a Kármán vortex street. It's a periodic flow pattern that's generated by the mountain blocking the flow, and basically you have vortex shedding due to the separation points vibrating from the left and right in an oscillatory manner. You see that same Kármán vortex street behind fish. So that mountain range I showed was on the order of kilometers, these fish on the order of 10 centimeters. And that's just amazing that you see common patterns, and that's basically a statement of that the forces are also going to be related to each other. That these two systems of enormously different scale are what we call dynamically similar. And dynamic similarity means a few things. One, that the wake patterns look similar, as you can see, but we can also be more precise in when dynamic similarity happens. And that involves not comparing the sizes of these things, but comparing the forces. And that's where dimensional groups comes in. So as Laura will show in the later part of the talk, none of these simulations make sense without having the context of the dimensionless group governing the system. So in fluid mechanics, we'll often talk about forces that arise in the fluids, and a dimensionless group is simply a ratio of those two forces, be it inertia forces, be it viscous forces, be it capillary forces, or others. And to calculate these forces, what we use is physical parameters. So physical parameters are basically mass, length, time. Time could be the frequency or the period. Velocities can be involved. And a system, the reason why dimensionless groups work, the reason why we can characterize, that Guadalupe Island and the fish, is because systems do not depend on the units of dimension. So it didn't matter if I measured that mountain range in kilometers or miles, or the awkward foot for the US customary system. What matters is the physics of the system, which don't depend on arbitrary chosen units of dimension. If you want to read a little bit more about the background of a dimensional analysis and the methods to find these groups, I teach with an undergraduate textbook, Monsignor Okishi has a whole chapter and practice problems that I think anyone can do. So dimensional analysis, we're trying to get these dimensionless groups that don't depend on the units of dimension, and it also helps us minimize the number of variables that we can use in a system. So if you want to look into the background of how to calculate these dimensionless groups, you can go back to 1886, where he was a soil scientist, Edgar Buckingham. He was trying to model flow through porous media, and he noticed that, hey, I could do these experiments at different scales, and they both seem to have related results. He basically reiterated results from Joseph Bertrand that is now called Buckingham's theorem, even though it's actually both their works. Buckingham's theorem simply tells you how many dimensionless groups you have. And luckily, most of your systems will have a handful of groups. It's not going to be, even the variables you can measure can be more than that. The groups usually are fewer. And that's also the benefit of using dimensional analysis. So Buckingham's theorem, it's derived based on linear algebra. And it basically says, if you've got, you want to find out the number of groups you want, all you do is figure out the number of variables you have. I'll give an example of this when we look at walking on water, and count the number of units, which are usually mass, length, and time. So usually it's 3 units. And well, the difference between the number of variables and the number of units is m minus n. That will tell you the number of groups you need. And the behavior of their system is uniquely prescribed by those groups. So if I wanted to model what's going on past that island or that fish, all I need to do is measure the dimensionless groups, which are coming from the variables. There's special cases, and you can look at this in the literature. There's some very nice problems in which you have a single dimensionless group characterized in your system. There's a famous story of Taylor, who's a fluid dynamicist, that was looking at the atomic bomb explosion pictures from tests of the atomic bomb, and he was able to predict the energy used in the bomb based on how much that plume expanded over time. That was a special problem when the problem of a bomb exploding is characterized by a single dimensionless group. And Buckingham's corollary says that group has to be a constant. But in general, the groups depend on each other. And so if you want to change your system or figure out some dominant physics, you have to calculate all these groups and figure out how they change upon each other. And that's usually from experiment. So that's the basically general idea of dimensionless groups and why we care so much about them. So I'll give you a few case studies where we actually calculate some dimensionless groups for some animals that you might know about. So let's start. This is actually worked on this with John Bush when I was a PhD student. Let's start looking at the problem of walking on water, which has evolved a number of times, from these birds. Like Clark's group can slap the surface and run on water. I'll show a video of the Jesus Christ lizard, and 1,200 insects and spiders have all evolved independently to basically, use their legs on the water surface. The videos I'm going to show you, this goes back in the day of me as a grad student. Here I am with my colleague Brian Chan, who built this first mechanical water starter. So for this walking on water problem, as well as the problem of the dog shake and the cat cleaning itself, those problems involve animals that are often much bigger than my finger, but they are all dominated by capillarity due to the small scale interaction between the animal and the surface involved. So the dominant physical parameter that we want to measure is surface tension. And I'll talk about some other parameters when we go into dimensional groups like density and viscosity. But for walking on water, surface tension is quite important. So surface tension has the units of 70 dynes per centimeter. Dynes is a unit of force, and centimeter gram per second units. And the important thing to know about that, it has a force per unit length. And force per unit length can be interpreted as also an energy per unit area. So imagine you've got a water surface. If you're really small, we'll show that basically you can jump on it like a trampoline if you're hydrophobic. And the weight support that you get is basically going to be, since surface tension is force per unit length, is going to scale as the length of your leg times the surface tension. So you take 70 and you multiply the length of your leg in centimeters, and then I'll give you the force. There's a subtle point. Because the bottom of the leg is wetted, you actually have two contact lines that actually support your weight. So there's a factor of 2 in the front that I'll show you in detail in a second. So just based on the units of surface tension, you get an idea that it's going to allow you to have weight support because it has a force per unit length. And let's go a little further with that. So there's these insects that slap the water surface to support their weight. For them, surface tension is not relevant. In fact, I worked with Jasmine Rody and Bob Full and showed that if you're really big, you can just add a bit of soap to the water and you can just slap the surface and support your weight just fine. If you get smaller and smaller, you're this regime, there's these small geckos that are actually in between the sizes of insects, which are, the size of your pinky, and the size of these lizards, which is the size of your hand, which surface tension is important, and they actually use both modes. But in general, you can think of, so you can have this dimensionless group, which is the weight of the animal divided by surface tension times the perimeter level. That's dimensionless because surface tension is force per unit length. And when you're really small, that number is really tiny, which means that these animals have a huge amount of weight support, and they can have a factor of 10 in safety. So we're going to go into weight support carefully, but let me give you the big picture for force generation. For example, this Basset's lizard, I said it supports its weight with slapping its leg. If you look into my annual review, the force on the leg is basically given by a couple different parts. And the important thing is really this line here, which lists the scaling of these forces. So necessarily before you do the scaling, you have to start with calculus, where you looked at the areas of these things, the contact lines, and any kind of curvatures. But ultimately, when you're looking at the scale of these animals, getting a scaling which involves basically turns integrals into products is actually sufficient. For at least the case I'm going to show here. So for example, if you're a Basset's lizard, you want to support your weight, you can't stand on the water surface, you've got to slap, and their form drag is important, where the density of the water times the velocity of the leg squared times the area of the leg will support your weight. That's why it doesn't work in air, because water has a density that's 1,000 times that of air. These lizards also need buoyancy forces, where the weight that supports a boat is density times gravity times the height that you are underwater times area. They also use weight support in that fashion. Added mass arises anytime you're accelerating a fluid, in particular when you've got air cavities accelerating fluid. So not only do they drive their legs downward, but they also slap it. So the water is initially stationary and it's accelerated. And this is the reason why if you look at rising bubbles, there was a paradox a long time ago called, I think D'Alembert looked at it, but basically bubbles rise. If you just look at form drag, bubbles rise a lot slower than you expect. And that's because of the influence of added mass. So you can consider it to be the fact that if you push an object through water, not only do you have to accelerate that object, which you know from Newton's law, but you also have to accelerate the fluid around it, because that has to go into the place that the object was originally. So you're basically pushing both the object and the fluid around you. And to do so, you have to consider the volume that's displaced, which is here, the density of the water, and the acceleration. Viscous forces generally are not productive for locomotion. They are generally dissipative and they generally slow animals down. So that scales of the viscosity times the velocity times area. And I already talked about the curvature forces, which goes to the surface tension times the perimeter of your leg, or that's similar to times the area of your leg divided by the width of your leg. And there's some special cases where insects can actually leave little drops of soap-like fluids, and they actually kind of rip a hole in this surface tension, and that caused them to fly forward. Those are called Marangoni forces. So I've described these forces in terms that you can understand, but generally when fluid dynamics is this, they basically they talk about things not in terms of forces themselves because those have units. It's very difficult to interpret a physical system just based on for example, I say it's got 5 newtons of form drag. Instead, it's much easier to talk about things like the Reynolds number, Weber number, Bond number, and Streamline number. Those are very common dimensionless groups, and those are simply found by taking, you can see I have about 6 forces here, and pairs of these forces lead to the ratios of pairs of these forces lead to these dimensionless groups. The most important one is the Reynolds number, and I guarantee if you're watching this lecture, you will deal with this Reynolds number. It is the most famous dimensionless group. So let's talk about that briefly before we go back into walking on water. This is a slide from Gianluca Dotti. The Reynolds number is the product of the density of the fluid times the speed of your appendage times the length of your appendage divided by a material parameter, which is the dynamic viscosity of water. So Reynolds numbers, air and water are quite different. Let's just talk about liquids. So the amazing scale of life, you go down to things that are microscopic, 1 micron, or even the cilia on those organisms, which are fractions of a micron, all the way up to blue whales, which are 100 meters. So that gives you 6 or 7 orders of magnitude in length. And the fact that Reynolds number is a product of speed and length means that you got basically 15 orders of magnitude across life on Earth for the Reynolds number. Laura will show you a lot of simulations of different Reynolds number, but basically we live at high Reynolds number. You'd have to basically be really small or going through molasses to deal with low Reynolds number. The higher Reynolds number is, the less predictable and laminar the fluid streamline patterns are. That's shown by this case. This is from Munsung Kishi that talks about how the flow past a sphere and fluid, or you can consider, to be honest, a cylinder would be probably better because it's 2-dimensional. It's quite laminar when the Reynolds number is low. So when basically the length scale is low or the viscosity is high, the Reynolds number is low. Laminar meaning each fluid streamline is basically sort of follows the other. I can draw them without having them cross over. As I increase the Reynolds number, I get separation. So I basically get these convective patterns that sort of stagnant areas in the wake that sort of, if I've got little dust things, they'll stay there. Intermediate Reynolds number, I get this vortex street that I showed you with Guadalupe Island, all the way up to super high turbulence Reynolds number, where you got a mix of both vortex streets and a mix of turbulence, which is not easy to characterize with the scaling or with CFD. So this Reynolds number is very powerful. Just by someone giving you a number, if I said the Reynolds number is 1,000, it's a very different world from a Reynolds number of 0.1. And so that's why generally when you give a simulation or you even characterize a physical system, that's really the big number here. So back to this dimensionless group of weight support. Let's apply it to a person walking on water. If you want to walk on water, well, the thing about these insects is that they've got really thin legs and they're orders of magnitude smaller in size. That means even smaller in weight. So for us to walk on water, if I'm 100 kilograms, I've got to have a perimeter of my leg that's about 10 kilometers in perimeter. So that's the basic reason why humans cannot use surface tension to walk on water where insects can. It's a basic scaling result due to the ratio of your perimeter and your mass. If you wanted to run on water like that basset lizard, you need to have legs that are feet that are about a square meter in size, which seems actually kind of reasonable if you had garbage cans as lids. But also that form drag requires you to move water at the speed of a person running. And unfortunately, that requires artificial aids because the power involved, which goes as the velocity cube, is much higher than you can get. You need 15 times as much muscle. So let me just show I had mentioned this basilisk lizard. This is it using those three forces I talked to you about: form drag from pushing its legs at a certain speed, acceleration from slapping the surface from the initially quiescent water accelerating, and hydrostatic pressure, which act on the bottom of its legs. It has this extra ability to pull its legs out before the cavity collapses, so it doesn't have to deal with as much viscous drag if it had slow legs. That's it in real time. Don't try that at home, you're gonna get wet. Here's the water starter that I had mentioned. This is a photograph that we took with John Bush, where you can see basically it's supported by surface tension here. So its weight is supported by these little, so this whole insect is the length of, it's a centimeter long. These legs are the width of your human hair. And those are great implements for supporting your weight because they got tons and tons of perimeter. Look at the perimeter of all these insects, which we'll calculate a little more carefully in the next slide. Those are supporting its weight, just like it's easier to balance a paperclip on the water than it is to balance a paper weight because of these thin implements that basically utilize surface tension. Here we showed that it pushes itself forward by a combination of capillarity and inertia. You see, these are the vortices, the same vortices you saw in the wake of Guadalupe Island. And that's because if you calculate the Reynolds number of these insects, there's about 100 to 1,000. If the Reynolds number, if there were microscopic water starters, it would not generate vortices because its Reynolds number would be much smaller. And the length scale of these vortices is dictated by the length scale of the legs and the fact that they're actually deforming, they're actually pushing down the water surface, generating little dimples of air that push down on water. So they use their legs as oars and they use the dimples, the little cavities, as blades to push themselves forward. This is the textbook actually that I end up teaching with at Georgia Tech used this water starter as a cover, so I was pretty happy to see that. So this is one of three instances that I want to show you how you can use the scaling to make useful predictions for biology. These are different. So I showed you one water starter, but the joy of our field is that there's so much diversity, and that diversity you can actually help quantify. So these are actually hand drawings and from Anderson's book in 1982, where he drew almost a couple hundred species of these insects that walk in water. And you can see they're going to have various gaits. This is clearly a rower. It's got really long rowing legs. This is more of kind of a cockroach-like tetrapedal gait. But because we know that the maximum surface tension forces, and this is the key about simplifying for these models, I know that the actual surface tension force will be a function of the contact angle theta, but the maximum force you can use to support your weight assumes a contact angle that's perfectly hydrophobic and that the force scales with the length of the leg times surface tension. So I can actually calculate the surface tension force for all these animals by measuring these leg lengths, which this is a great job for beginning grad students, undergrads, to basically measure this. And then I can also estimate the weight by assuming their body's like a cylinder, and that is also the density of water, which you can confirm that it's pretty good by actually weighing some of these live insects. And then I can construct this, we used to call in grad school a mega plot. So a plot that combines hundreds of anatomical drawings and gives you physical intuition for the system. This y-axis is the surface tension support force. This x-axis is the weight. Now, this line here is when y equals x, basically when the weight is equal to the surface tension force. If you're below that line, you've got bad news. It means that you're not going to be able to support yourself with surface tension. And that's why none of these insects are below this line. And the vertical distance that you're above this line tells you your safety factor, tells you how much extra weight support, how close am I to basically falling through. And all of these insects have safety factors of 10, but you see the amount of safety factor starts decreasing as you increase in size. The smallest safety factor of which is that point on the top right, which is I've drawn in the inset here. That's this giant Vietnamese water starter. Seriously, there's an insect, that lives in Vietnam in the rainforest that has a body that's, 4 centimeters. So that's 4 times as big in length for the body. And its legs, that's actually one of the pictures from one of these books that document it, are almost as tall as my head. So that insect is really playing with fire. It's a safety factor of 2, which allows it to simply perform the task of mating. But otherwise, that has really pushed the limits. We can have no water strider, no water walking insect that's larger than that, unless we were to play games with not making it out of water or basically using other means of propulsion like slapping your legs. So it's a very useful, so this, you can see the scaling and understanding the importance of surface tension and is useful because you can incorporate all these data from the 1960s, 1980s and get some new insight into evolution. So that's all I want to say about water starters. I want to talk about some animals that you probably work with every day. We all just got back of the COVID epidemic. I think the only reason some of us survived was the cats and dogs and animals in our lives. This is a dog shaking water. It's a fascinating thing. I mean, this is a high-speed video of them doing it. It's a problem of surface tension, and it's a problem that actually you can solve without looking too much into how different these animals are. Here you can see it started with its head, it's trying to get the water out of its ears, and if it's really good, it also do a little twist with its tail. So how much water do animals accumulate in their bodies? This is also a problem of scaling, and this is a Haldane brought this up in the 1920s and has a great essay on being the right size. We corrected some of these, his actual measurements. He said correctly that if you come out of your bathtub, you have about, you don't use a towel, you've got a thin film of water on your body that's a 50th of an inch thick, and that weighs roughly a pound. A rat comes out of the same bathtub and it has 5% of its body mass, and an ant has 3 times its mass that it carries around. And that's due to the increasing surface tension to volume ratio. Basically, when you get smaller, you've got a lot more surface area, relative surface area to deal with, and that has consequences for drying. Imagine if these animals are in winter and they want to basically, if they gotten rained on or if they jumped into a lake, it could be death for them because water, if you ever saw it around in wet clothes, you have the luxury of taking your clothes off, but animals don't. And a 60-pound dog would have a pound of water in its fur. There's a heat of vaporization that is required to basically turn water into vapor. And a pound of water would use about 250 calories of an animal's daily energy intake to burn off. So it would have to use lunch to basically dry off if it did not evolve this wet dog shaking mechanism. Because of the consequences energetically of keeping the water on, all furry animals had to evolve this method to remove the water. This is a cute little mouse, and as with many things get difficult the smaller and smaller you are. The dog I showed you in the first slide, that husky, juvenile husky, is shaking about 4 to 8 times a second, but this mouse is about 27 times a second. Here's a rat, it's bigger than the mouse, so it's about 20 times per second. Human eye blink is a third of a second, so you get 6 of those in with every blink. Notice it actually closes its eyes when it does the wet dog shake, and that's because, as I'll show, we can calculate the gravities involved. So the amount of acceleration exceeds what humans can take. So in a typical car crash, you get 5 to 10 Earth's gravities. That's called G's. And this rat exceeds that, so it's got to basically close its eyes so its eyes don't end up popping out of its head. Colonel John Stapp showed that. He said he actually self-tested, put himself on a rocket sled, and he showed that after 10 G's, you get retinal detachment, which unfortunately he suffered through for a lot of this. Here's some shaky bacon, pigs shaking off water. We expected sheep not to shake because they have lanolin, so water repellent animals like otters and beaver. We thought they'd have other ways to prevent water from aggregating, and they do, but still they've got to deal with removing water. I would say this is actually quite artistic. I think this would go on MTV. This is a little dark maybe on your screen, but this is the largest animal that was available to us. We filmed pandas and bears. This is videos from BBC showing bears shaking. It's so vigorous it actually can use the inertia of its eyelids to pop them open. So I mentioned in the very beginning, you start with a simple observation, and nothing is simpler than just putting water in an animal and measuring the frequency they shake. That basically starts paving the way to do some scaling. So this is the wet dog shake rule, where you've got 32 animals where the frequency, the number of cycles per second of their shake is on this axis, and their body mass is on this axis. And as you saw, the mice and the rats were the fastest. Then you basically get the slower animals all the way up to black bears and brown bears, which are only shaking at, half a cycle per second. And the domesticated dogs are all below what you expect from these sort of wild animals. They sort of worsen their ability. And as I'll show you with the work we do with cats, domesticated cats also can have some issues with cleaning themselves. But all these animals tell you that evolutionarily there was a trend. There was basically there's some gene that tells these animals how big to make your bones, how strong to make your muscles, how fast you move all this to get this shaking frequency. And as you can see, water was throwing off all these animals because it was effective for all of them. So why do these animals shake at different frequencies? Why don't mice just shake like a dog? Why they got to do 30 times per second? So to get into the modeling, we've got to do a little bit more careful experiments. And this is Andrew's Labrador retriever at his backyard with a pink straw taped to the back of the dog. So that is the back of the dog. So if I were to do this same experiment with myself, I would not get that much motion. And this gets to, a long-standing mystery in animal movement. Why do mammals have such loose fur? That back of the dog is going from, as I said, if I did it to my back, it would go 11 o'clock to 1 o'clock, but that's going from 3 o'clock to 9 o'clock, almost 3 times the amplitude. And that's what you can see here. It's got this huge amplitude, more than you expect based on the vertebrate motion. So this experiment gives us an amplitude of the shake that's going to be important in modeling. So based on the higher amplitude with a similar period, you're going to get a higher force. In particular, we'll be looking at centrifugal forces, which are going to scale as the amplitude and the frequency. So basically, if you multiply, the centrifugal forces go with the velocity squared. So if you square the velocity, you're going to square the amplitude, and you're going to get, if you got 3 times the amplitude, you're going to get 10 times the force. So it's incredibly powerful, increasing the ability to shake water. I mentioned that my backbone doesn't move very much when I shake. It's really mostly the loose skin. You can really see that with, this is Young Hee Chang's X-ray of a wet rat shaking. And you can see that backbone is really stationary. It's really the skin that's moving around the body, as you saw with the dog. So this is a video when my wife and I were dating. Actually, that's actually my wife's ex-boyfriend's Valentine's gift, a poodle. And it shows, the poodle's quite scared, but he's okay. We chopped a little off its fur to basically see if we can understand what physical processes are going on when you're shaking at 30 times per second. And we actually put a high-speed camera in the same view of the hair. So this is actually, if you're a camera on the dog looking at what happens. And you can see it doesn't matter how big the dog is, the process of losing a pound of water boils down to drops leaving your body from hair clumps. And the forces involved in that process are as I said, imagine you basically have water that's attached to you. That water drop has a perimeter of R. As it's being shaken, if frequently you go to the bathroom, there's no towels, you got to shake your hands. Gravity only has so much acceleration, so you amplify that gravity by accelerating your hand several G's. That gives you this inertia that's pulling this and pulling it off. So there's those two forces that are sort of at the interplay. If you incorporate animal sizes with that, the fact that basically the bigger you are, the bigger your mass is, your radius doesn't go up with a cube, it goes up with 8/3 due to some other anatomical reasons. You can basically calculate the centrifugal forces, which goes as the mass, the drop, the radius of the contact, frequency squared times amplitude squared. And those are basically fighting surface tension forces, which goes the radius of the drop times surface tension. So these two together give you a frequency that's a function of radius of the animal, which you can relate back to the mass of the animal. Frequency goes as mass to the -0.19. That's a testable hypothesis that, as I showed in the beginning, let's go back. Show that that's this theoretical curve here. And without any free parameters, just by doing experiments with my wife's poodle's fur on that spinning device, we're able to basically understand that smaller animals have to shake faster because they have a smaller radius. To compensate for their smaller radius, they basically have to increase their frequency to exceed the minimal force that it takes to remove the drop. That means it's a function of what's going on in a small scale. And as a result, these animals have to have huge numbers of gravities of acceleration. But because they all want to just exceed the forces to remove the drop and not go any further, they're generally within this range of 20 Gs to 60 Gs. It also tells us that animals have been tuning their bodies to remove water that's trapped on their bodies due to adhesion by surface tension. If you're trapped with oil, if your body's covered with oil, the dominant forces, so in that case, are not going to be surface tension that holds onto your body. In terms of dimensionless groups, we're going to go to low Reynolds number, and basically what's trapping the body, the fluid on your body, is now viscosity. And because you're shaking at 30 times per second, you can remove surface tension, remove drops with surface tension, but you cannot remove oil. So these animals that are oil covered from oil spills that will unfortunately still be happening for many years to come, They cannot clean themselves on their own because their methods have been tuned to the properties of water and not for oil. I've got one more story to tell about cats lapping, licking their bodies. And this is Alexis Noel's PhD work. Cats are a great species to study. They've got a huge range of sizes. And one thing they all have in common, they love grooming themselves. It evolved partly, a lot of due to parasites. There's been a long history of parasites of mammals. Cats have fleas and ticks. There's a flea and tick burden. And to maintain those number of parasites at a low enough value that they can grow and develop, they have to be able to remove these things, and they do that methodically by using their tongues. This high-speed video of that action. So again, like I said, we start modeling with some simple experiments, and it can't get much simpler than trying to watch a cat lick itself. I mean, I say that, but in actuality, this was actually, even though it looks like a simple experiment, these simple experiments take sometimes lots of planning. For example, this video here, I understand it's probably a little choppy on your end because I'm using Zoom and PowerPoint, so I'll play this a few times. This is actually a cat licking itself on the left, but to get a real good view, we had to get a cat licking a carpet covered with cat food. So it actually took a little bit of work to see the action that the spines actually have a pretty active role. They're not just passive, but the cat tongue expands like a balloon, and you have this interaction between the spines and the fur. Don't worry, all those cats I just showed are not harmed in the study. We were able to obtain cat tongues. So they're house cat tongues about the size of your pinky. Then you've got a bobcat, cougar. That lion tongue's about as big as my head. And that just shows you that this method of cleaning is really, is lasted, and as these predators got evolved bigger and bigger sizes, it's really stayed the entire time. The tongues are not particularly long for their size. Basically, what we show the length and width of these tongues is isometric, so it's as you would expect. The aspect ratios do not change as the body size increases. So in the literature so people in the past, they always thought the things on cat tongues, they were just spikes, cones. With the advent of 3D scanning and 3D printing, you can actually go much further. And what looked like simple spikes on SEM, as I'll show you with this 3D scan and a 3D print of cat's papilla, those are those small spikes that you see, that there's some really interesting geometry here. And to do this physical modeling, it's really important that you get an accurate picture of the interaction of the two surfaces. So you see there's a kind of like a skateboard ramp inside the papilla. It's a hollow ramp, and to be honest, we observed it by accident. Alexis was scraping a steak with a cat tongue to see how they scrape meat off, and she found she couldn't clean the cat tongue. So she realized, hey, there's actually some really small structure here. Somehow this structure is preserved across cats, even though I would much rather see my own house cat in a dark alley than a lion. If you're licked by each of those animals, the pain level is probably going to be pretty comparable, because the length of that lion papilla, that region, is the same as a house cat. These are all these papillas from all these different animals to the same scale. So this leads to an interesting question. Why, if so many other parts of animals increase in size, why did that papilla stay the same size across animals? And this is a question that you can answer with a combination of basically measuring things from museum specimens and from doing some modeling. We went to American Museum of Natural History, and they have a sort of a lush sort of closet of tons and tons of furs, where you can measure the density of furs, so the number of furs you have per volume, the radius of the hairs, and the length of the hairs. And when as you saw from those videos of cats licking themselves, when they're licking, they're compressing the furs down to a minimum height. To measure that, you can actually model that minimum height by keeping in mind, if you've got a cylindrical lattice, the minimum packing density you can ever get if they're packed is 0.09. So basically, it can go down to compress down to 10. Basically, it's 10% of the volume is due to the fur. So based on that idea, this idea of this epsilon for this dimensionless compression, based on the density of the fur and the hair properties, you can estimate, given how long a hair is and how many hairs there are, how much that would compress down in terms of a baseline height. And here's that calculations of the baseline height for all these different animals. And you can see there's and this is the base of the height of the cavity, which, as we saw, all those animals have the same height of the papillae. And you can see that you kind of get two regimes, that nature has basically designed all the hairs and the densities so that the animals can groom them. so that the height of the papillae can penetrate through the compressed fur. Even animals like the snow leopard, which have incredibly dense fur to survive winters, they can still groom themselves. Animals like this adorable Persian cat you see here have fur that has gone beyond what their tongues are able to handle. And that's because Persian cats did not evolve in the wild. We basically bred them to have fur of such length and density that Persian cats actually have difficulty grooming themselves. If you want to own a Persian cat, you're going to have to basically bathe it regularly, dry it carefully, and brush it thoroughly every single day. So, or you can just shave the Persian cat. But if you're going to do that, I would say, hey, just get a Chihuahua. So Persian cats really are not able to groom themselves, and we can show that physically based on their hair measurements and the height. So we've shown that geometrically the tongue can penetrate, but what is the physical process of grooming? Every time we've observed that cats will take about 10 sort of grooms of a place before they move on to a different location. And what's going on in that process when they put their tongue back in their mouth? Well, the glands of their tongue actually generate saliva. And as you can see, this is a high-speed video of a drop interacting with the papilla. And the papilla is made of a hydrophilic material, and that cavity is of a length scale that you get automatic wicking. Wicking is a process that happens anytime you've got hydrophilic materials and small porous cavities interacting with water, where you're at a length scale that gravity no longer matters and water is automatically embedded into these surfaces. You can calculate the speed at which it's embedded by Washburn's law, where basically you've got capillary force driving it and viscosity resisting it. And that's why it takes on the order of like fractions of a second for this to happen, because the capillary force is so strong. And that works well to describe the drawing forces of each of these papillae. So this is kind of a gruesome experiment, but don't worry. These cats were frozen. They're from Tennessee tiger heaven. That is a cat tongue that we can basically start to do real precise experiments where we can test these hypotheses for how much water gets pushed into the fur, how many grooms it takes to clean an area. So we basically built this automated grooming device. We have a cat tongue, and we can measure the weight of the tongue before and after to measure how effective it is. And the big physical picture we see is that cats are incredibly efficient at removing dirt from themselves and using water. When you take a shower, you go for a quick 5-minute shower, it'll take maybe 10 to 20, 50 liters of water. It's a huge amount of water. Cats, they cannot afford to use that much water on a daily basis. So they have evolved as a mechanism to put fluid just on the individual hairs. So it's like you're basically just bathing each of these individual hairs one at a time. And they do that through their 280 papillae interacting with the hairs themselves. What you're seeing here is a single hair being pulled through that cavity, and it's getting a small bath. And because it's releasing just the amount of water to coat a single hair, there's no waste. So it's basically just dripping. Basically, we calculated based on measuring the weight of these tongues and the number of grooms that cats can have in a day. And they spend about 16 hours a day grooming, which is ridiculous, that a cat uses about 3 tablespoons of water per day, whereas we use 50 liters. And they're actually quite clean. The method is so effective that of basically putting water into their fur, that cats, they've also lost their sweat glands. And to basically cool themselves down, they'll just lick themselves. And you can see from this thermal video that the hairs get cold as the water evaporates. I think that's as far as I want to go.
Does anyone have any questions right now?
David,
Okay.
I have a question for you.
Uh-huh.
I was kind of wondering how the material or just the different characteristics of the fur, like density or follicle stiffness or length, how that comes into play with the shaking off water behavior and the scaling relationship you mentioned earlier.
That's a good question. We clumped hair into different, so the process of clumping is actually very complicated. Just like when your hair clumps out of the shower, you pick up those gooey hair clumps from your bathtub. The process of how big the clump is a function of, like you said, the density, the space in the hairs, and the elasticity of the hairs. And the clump size will dictate sort of the substrate that the drops leave. In particular, if you don't have clumped hairs, I think it's really, really hard for drops to escape your body, because you got really thin layers of fluid all over each individual hairs. So I think clumping, which is a function of elasticity, does help the animals remove the water, but we did not study that systematically because you'd have to measure the elasticity of each of these individual hairs. Moreover, the process of, so there's actually, in actuality, the mammals have both over the guard fur, which is stiff, and the under fur, which is sort of soft and fluffy and nice to touch. The interplay between those and where water is actually trapped is complicated. So the method I've shown are really good for figuring out basically the big question of why they're shaking. Basically, they're shaking faster when they're smaller because they need to have a higher frequency to compensate for the radius. But the subtle questions of why, like how water leaves fur, that might have a result in, like as you saw, it was not a perfect scaling. That might have some consequences in how wet they stay after the water's removed, and with any kind of particular like mechanisms they use. And there's definitely a lot of like animals, like from water animals to desert animals, that I think there'd probably be a lot of things to study there. So I think there's some fascinating questions with how fur evolved to deal with water that could be addressed by measuring.
Great, thank you.
I'm glad this was useful.
Okay, I don't see any questions. All right, so let's see, we can pivot to computational fluid dynamics. So I'd like to talk some about detailed models requiring numerical simulation, but as you might imagine, covering too much in 50 minutes when it comes to CFD is, well, not really feasible, but what I want to do is to provide an overview of the various tools that are available and talk a little bit at a high level about the process of doing simulations and to offer some tips and warnings as we go along too. And I would say definitely in the last 20 years, there's really been an explosion in terms of scientists and engineers using computational fluid dynamics to understand fish swimming, insect flight, bird flight, and other problems in biological fluid dynamics. This is a nice example of a very collaborative paper in Nature from Bonfrey's group looking at the flows produced by a mosquito wing. And let's see. So I want to give a little bit of encouragement right now as well. If you're a biologist who hasn't been trained in numerical analysis or partial differential equations, that there are a number of people who have made that transition into doing CFD. And so these are just a couple of examples. Lindsay Waldrop, who's at Chapman University, has been doing immersed boundary simulations of flow in tubular hearts with inspiration from the sea squirt. And she was actually just awarded an NSF Division of Mathematical Sciences grant in collaboration with Yan He. So she has been extremely successful in using immersed boundary methods to look at sort of comparative biomechanics of tubular hearts and ascidians. Eric Teitel, who is doing our part 2 workshop and highlighting some of his experimental work, has also been involved in a very successful interdisciplinary collaboration looking at lamprey swimming, again, using the immersed boundary method. And that group has a number of papers looking at sort of coupling between activation and muscular mechanics, movement of the lamprey through water, and feedback. And then Jake Soha has collaborated with a number of different people in fluid dynamics, doing CFD models and more analytical modeling as well. And I'm going to mention a pretty interesting paper that highlights some of his work. That this is a cross-section of a gliding snake. So I'll mention that here in one moment. And just to provide a little more encouragement too, if you choose the right problem, then you can also engage undergraduates from all different fields, math, biology, engineering, to use CFD to look at flows around and through organs and organisms. And these are a couple of examples. This is flow over a horseshoe crab, which was done by Alex Davis, who at the time was an undergrad at UNC. And he did a 3D scan of a horseshoe crab and then performed immersed boundary simulations looking at lift generation over the shell. In addition, I have a class that's for, well, there's been a number of iterations of this class, but I've run it as a first-year seminar in biology, as an upper-level math course, as a fluid mechanics class for BMEs. But in all cases, I have had groups of students use ANSYS and COMSOL to perform simulations through organs. So this group made a preliminary model of the embryonic heart and then used ANSYS to look at flow through that heart. All right. So there are some challenges, and like I mentioned before, about making the correct modeling choices so that what you're doing is computationally feasible is important. And if you think about fluid structure interaction in the natural world and how, as biologists, one might think about it, it's great to appreciate all the complexity across different scales. And I have some examples of pumping. So this is a whale heart, actually, at the large scale or the small scale, where we have cilia in the respiratory tract. And then also at the large scale, thinking of locomotion, stingrays, all the way down to E. coli. And so your first thought of this is that, wouldn't it be great to incorporate all of this complexity, all of the morphology of the stingray, all of these cilia, hundreds or thousands of them packed closely together, all the variation and complexity. And one could definitely continue in a path where you're adding more complexity and looking at those nuances. But you probably do not want to start there because it will be extremely difficult. The other end of this is thinking about how numerical analysts often think about fluid structure interaction, particularly the mathematicians who are developing fundamentally new methods for solving these problems. And in numerical analysis and scientific computing, this is probably, I would say, the best studied fluid structure interaction problem. It's a rubber band in a 2D fluid. And so you basically just have this closed rubber band. It resists stretching. There's some volume of fluid trapped in between. And it's pretty easy to code up, right? You just make a circle, you add some springs between your vertices, you deform it initially, and then it wobbles and eventually settles down into a circle. And if you're approaching it from that viewpoint, you might be asking, okay, how do we take this example and make the simulation more accurate? How can we speed things up? How can we do a better job conserving volume? So there's a trapped volume of fluid here. And in a lot of methods, some fluid will sort of squeeze out or you'll lose volume in between. So how do we conserve volume, and how might we look at this problem in a totally different and better way? So we have these sort of two ends of the spectrum, from simplicity, a mathematician's spherical cow, all the way to thinking of the great diversity and complexity of organisms. So what do we do as we move forward? And depending on where you are in the spectrum, you might make different choices in terms of the software that you use. And so there's a lot of different software available. I'm going to mention some of the more popular ones, although, it's a little bit specific to what I do, just what I'm familiar with. But you do have a choice if you're going to look at open source versus commercial software. And there are advantages and disadvantages to both. You can also write your own. And if you're a numerical analyst, that's often what you might do. But if your ultimate goal is to move to 3D simulations with a lot of complexity, maybe multi-phase fluids, complex biological structures, it's hard to write your own code that goes all the way to that. So some of my collaborators have done this. Boyce Griffith is one, and it's probably taken him 20 years to get his software up to where it is now. So again, it depends a bit on your goal, but let's say you're going to start by looking at other software that's out there. All right, so I would say OpenFOAM is, at least in my experience, maybe the most popular open source CFD software. And again, if anyone knows of others, please feel free to add them to the chat, and I'm happy to add them to this PowerPoint as well. So OpenFOAM, you can do 2D and 3D simulations. You can install it on Linux, Mac, and Windows. There are a number of tutorials available on YouTube. You can pay and have additional professional tutorials and help. And in terms of open source, it's relatively user-friendly. I have not used it extensively. I think it's not as easy as something like COMSOL and ANSYS, where you have this very well-developed GUI and the company provides quite a bit of individualized support. But again, it's open source and free, so that's a huge benefit. Another open source software that's out there is IB2D, which was developed by my former grad student, Nick Batista, and I'll talk more about it. So the nice thing about this is, well, it's only 2D, but it works in a MATLAB and Python, and you can relatively easily do fully coupled fluid structure interaction, where you have an elastic boundary representing your fish or your wing that deforms due to the fluid forces and then also applies a body force to the fluid. If you want to go to 3D and do the immerse boundary method, there's IBAMR. And I'll talk more about this in a moment too. And this was developed by Boyce Griffith. It uses adaptive mesh refinement. So as you move from 2D to 3D, things become computationally much more expensive. And so you have to have a lot of consideration for speeding things up, again, by using something like adaptive mesh refinement or having a well-parallelized code. This software is in C++, and it uses about, I don't know, 10 or 12 third-party libraries. One of the hardest things about it is installing it and getting all these libraries to talk to each other. But once it's working, it's quite nice for doing 3D. There is also QIBM, which is in C++, and is from Lorena Barba and has nice tutorials as well. And so these three immerse boundary codes that I've listed here, you probably want to focus on Reynolds numbers of 1,000, maybe 5,000 or less. So you probably do not want to be doing simulations with full turbulence. You need to have some turbulent model of the fluid for that. And if you're doing flow that's very, very small scale, so you can neglect inertia, you might use something different like method of regularized Stokeslets. But there is not a fluid solver that works across all ranges of scales. So depending on what scale you're at, you're going to want to use a different fluid solver. And I'll talk more about that in a moment as well. Now, if you want to go the commercial software route, here are some options that are available. So COMSOL multiphysics is a big one. You can install it in Mac and Windows. ANSYS is also a really nice package. It only runs on Windows. And here are some links for these. So if your university has a license for one of these, then you're in business. Otherwise, it's quite expensive, although there are discounts for academics. There's Autodesk CFD, which I have not used before, and SimFlow, which I've also not used. But the nice thing about these is that, they're packaged into a nice user-friendly interface that takes you through the whole process of doing CFD. And there's a lot of support, so you're paying for that support. Some of the disadvantages, if you're doing collaborations, it could be that your university doesn't have any of these licenses, or that your collaborators don't have it. And so that can make things tricky. And if you're, looking in terms of education and trying to share lesson plans and resources, then that's also somewhat limiting too, since a lot of institutions do not have these licenses. So it's definitely a trade-off. But I'll show you a little bit of both of these here in a second. And so I also want to quickly mention or address whether or not it matters. So if you use any of these software packages, are you going to get the same result, right? And so naively, you might think, well, all these are well-tested software packages that are solving Navier-Stokes equations with boundaries. And well, it turns out that you might not get the same result. And there's a really interesting paper by Lorena Barba that goes into reproducibility and replicable CFD and overviews how it's harder than you think. And she focused this paper on the aerodynamics of snakeflight problem that I mentioned Jake Soha had worked on. And she had used QIBM to do these simulations. And they went back and tried to perform them in IBAMR and in OpenFOAM to see if they would get the same results. And eventually they got mostly the same results. But just quickly, some of the issues that came up. So this is using OpenFOAM, and there were problems with how the fluid domain was meshed so that you ended up with sort of a spurious vorticity. And I can't remember the details of how they fixed this, but definitely creating your mesh where here's a zoom in of that, and at each of these nodes, you're solving for the flow velocity and pressure. And so there are challenges.
Little speck things that you see? Is that why you say it's spurious?
Yeah, so this is artificial, right? It's like positive and negative vorticity that's at really large levels. Sorry. To these elements. So that shouldn't be there. And if you zoom in, you can see sort of this noise, you could call it, compared to this simulation over here.
Okay, thanks.
Yeah, and I think, if I remember right, that this might have been some issue with using Gmesh for meshing, which is a free software program, but I'd have to go back and look at the paper for the details of that. But another issue that comes up is how you pick your boundary conditions. And so boundary conditions are always problematic. Of course, you're not, there's some finite domain where you're solving the equations of fluid motion, and then what happens at the boundary. You could imagine a wall condition. So it's like your numerical experiment is in a box, or maybe you want to have periodic boundary conditions or some such thing. Different solvers are going to be limited in terms of what boundary conditions are available. And in this case, you can see this nice vortex wake behind the cross section of the snake. And there are issues at this boundary here that caused changes in the lift and drag that were measured. And they were eventually able to fix it, but in their initial attempts, these issues came up. So this is mostly just a warning that for a lot of reasons, when you simulate, you shouldn't just trust that it's going to work, or that if you do it in different software packages and with different boundary conditions or meshes, that you'll get approximately the same thing. Okay, so now I'm going to delve in a little bit more deeply into various methods to solve these problems and tell a couple of historical stories along the way. So one of the first is the immerse boundary method, which was developed by Charles Peskin, who's a professor of math at NYU. This method he created in his dissertation back in 1972, and it was to look at flow through the human heart. And so this is one of his older simulations, and you can see the valves of the heart, and it's a very complex 3D simulation. I think this simulation was from the 96, I want to say, but he was, I think I'd be correct in saying, one of the first person who could really do this type of simulation with fully coupled fluid structure interaction, where you actually have a boundary that is elastic and deforming based on the aerodynamic or hydrodynamic forces, or hemodynamic forces in this case, that also applies a body force to the fluid and moves it. But as some encouragement for how biologists can get involved, so Peskin actually has his PhD in physiology. He started an MD PhD in New York City and was an engineer as an undergrad and was interested in creating mathematical models of things that he was learning about in medical school. He ended up dropping the MD and just doing the PhD and making his way down to the Quran Institute to learn how to come up with numerical methods for these types of problems. And I guess to connect things to another piece of the story, so way back when, I was initially a grad student in zoology at Duke working with Vogel, and I also became more and more interested in mathematics. And I talked to Steve about it and said, I think maybe I like math more than experiments, although I still do like experiments quite a bit. And Steve said, well, I heard about this guy, Charlie Peskin. I used to think that mathematicians couldn't help very much with our problems. And then I saw him give a talk and he changed my mind. And so I thought, well, this would be a good person to work with. So I did go to NYU and work with Peskin as well. So there has been this history of interaction and transfer between mathematicians and biologists and engineers. And so making these steps is not as hard as you might think. It's not easy, but it's definitely doable.
I love that story. That's such a Steve Vogel thing to say.
Yeah. Okay. So like I had mentioned with the immerse boundary method, we'll say the immerse boundary approach to fluid structure interaction. So these are challenging problems basically because of the coupling. And you'll see this theme come up as you try to couple different physics or different scales or different fluids. This will always create challenges. And so, you know, we're pretty good if you just have a box of fluid of solving for the velocity and the pressure at each of these nodes. But now let's say that we have this immersed elastic structure and we're solving elasticity equations on that structure. And we need some way to couple these equations that are also being solved on different grids. So the immerse boundary approach offers one way to do this. Again, I won't go into the details, but just to say it adds quite a bit of additional complexity. And it also makes the simulations more challenging and take longer to run. And this is also going to be true if you're looking at problems, multi-phase fluid dynamics with air and water interface, or non-Newtonian fluids like mucus, or if you're looking at heat transfer in a fluid and maybe the heat is being generated on the boundary. All these things will add complexity. One of the nice things about the immerse boundary method, since it has been around for so long, is that there has been a lot of work to add additional functionality. So I think each of these points was its own dissertation by various students. So you can imagine, while we have this elastic boundary, initially it was massless, or you assume that it was a neutrally buoyant boundary. But if you want to add additional mass, there are ways to do that. If you want the boundary to be porous, either an infinitely thin surface or a layer. If you want to think about creating mathematical models of muscle that have the right characteristics, the right force length and length tension curves, force velocity and length tension curves, there has been work to do that. And in a lot of the software packages, that has been implemented so you can use that functionality. So I mentioned IB2D, which is the MATLAB Python 2D code for the immerse boundary method. Nick Batista here is the developer. It is on GitHub. And now I am going to attempt to switch to my other computer and see if I can just show you. So I think, at least for me, seeing someone else run a simulation makes it sort of lowers the activation energy for doing it myself. So let me share screen. Okay, so I have MATLAB open, and basically what I have done is to just go to GitHub and download IB2D. And within it, you'll see Mat IB2D and Python IB2D, or Python, yeah, IB2D. I'm in MATLAB, so I'll open this. There's a folder called IBM Blackbox that has functions for different solvers, but you can initially just ignore that, put it in a black box, and then go to the examples. So there are a lot of different examples that are available. I'm just going to pick the easiest one, which I mentioned before, which is this rubber band problem. So I open it up. It's the example standard rubber band with springs, so it just resists stretching. And to run it, I'm just going to type main 2D. All right, and like I mentioned, so this is a rubber band where the resting length of the springs that make it up have a 0 resting length. So it would like to contract down to a point. but it has this volume of fluid that is trapped inside. So the rubber band eventually wants to minimize its circumference, so it will become a circle. It started as a ellipse. And then I can see, well, here's the boundary, here's the vorticity, the velocity field. This simulation is under resolve just so it runs fast, but you could, if you were doing real research that you wanted to publish, you would increase the resolution and it would take longer to run. But another thing that it does, it outputs the positions of these points as VTK files. Omega is the vorticity, pressures, velocity. So we saw the movie just to get to see what was happening. Okay, the simulation's working, but if we want to go back in and analyze it, all the data is available as VTK, which can be analyzed with ParaView, which is free software for data visualization. And I'll talk a little more about ParaView in a few moments as well. Okay, so now let me switch back to my other computer. If I can figure out what happened to the stop. There we go. Stop share. Okay. All right, and here is just a screenshot of that GitHub site. So if you wanted to get it, you could go to code and just download. If you're used to using Git for version control, then you can clone it. And then when updates are added to the repository, then you can easily download those as well. Okay, so Nick has provided some video tutorials, which you can find here. So it goes over the immerse boundary methods, some of the models, and also how to visualize the data. And there are a few papers from his website that go through sort of how to use it as well. And there isn't, well, so you can look at the various research papers, but there's not a user manual, which hopefully there will be at some point in time. And just as an example of what you can do, so you might be thinking, well, this is a 2D simulation. It looks relatively simple, but there are interesting things you can do. This is a 2D jellyfish. So this hemiellipses are jellyfish. And you can look at what happens as you change the Reynolds number. So our jellyfish at Reynolds number 4 started about here. And so if you're at a low Reynolds number, there's a reversibility or reversibility of the flow. So when the jellyfish contracts, it moves forward, but when the bell expands, it moves backward. And so at lower Reynolds numbers, the jellyfish doesn't really get anywhere. And then as a comparison, if you bump the Reynolds number up to 32, it actually swims and you can see this nice vortex wake. If we were to plot the average forward velocity versus Reynolds number, you can see this is a log scale. So at Reynolds number 1, there's very little net movement. And then once you get up to 100, it plateaus. And this is for a prolate jellyfish, oblate. This story is a little more complex, but this example is actually in IB2D. So if you wanted to simulate these jellyfish, there is a code for doing that. All right, so IBAMR is a 3D immerse boundary method. This is the adaptive mesh refinement. So the grid is refined near the jellyfish in the regions of vorticity, and it's coarser farther away. And it ends up that you really need this to make these types of simulations feasible. So otherwise, let's assume you had a uniform grid. You're resolving the fluid everywhere in this large domain, and suddenly, well, this simulation, I think each pulse on a cluster took about 12 hours. If we hadn't used adaptive mesh refinement, it might have been a week for each pulse. So it's really easy for these types of simulations to become intractable. And this code was developed by Boyce Griffith, and he started to work on it around the year 2000. And I think it was probably about 2010 before other people were able to use it. And now quite a few people are, but it's been 20 years. So like I mentioned, if you're writing your own 3D code from scratch with adaptive mesh refinement, it can take quite some time. All right, so let's assume that you are using IBAMR. So what is there and what do you have to do? So IBAMR handles the force spreading. So if your boundary is deforming, it's like a fin or a wing and it's applying a force to the fluid, the IBAMR handles that component. It solves the Navier-Stokes equations and it moves the boundary at the local fluid velocity, or it moves it with the flow. Your job is to create the biological boundary, which means making an elastic model, maybe with beams and springs, or a continuum model. You have to make the geometry and create the mesh, which can be quite challenging. And you might add some other features for how to describe the boundary using biochemistry, muscle mechanics, preferred curvature. So this component of the loop, you're writing your own code, you're doing your own modeling. So there's a lot of math modeling that's required. So it's not just black boxed. And this is just a quick example of what you can do with it. So we're moving from my 2D jellyfish to a 3D jellyfish, which required going from something like IB2D to IBAMR to make the simulations feasible. So we take something like a moon jellyfish, which is relatively simple for macroscopic organism, but we simplify it even more. We ignore the tentacles and oral arms and so forth. So here is our model of a jellyfish. And here are the results from IBAMR, and the jellyfish is making a turn. That simulation, I think I want to say, took 2 weeks on our cluster. So these are pretty challenging simulations because you're resolving the full fluid structure interaction problem, where this is elastic bell that's deforming as a result of the fluid forces and then applying that body force to the fluid. And we did not prescribe the motion of this turn two, I should say. The whole movement emerged from applying active tension that represents the muscles that deform the elastic jellyfish that then applied a force to the fluid and so forth. Okay, so that's a really quick overview of the immerse boundary method and the software that's available. And now I want to pivot just a little bit and show you some examples using commercial software. And so let's say that we're not necessarily considering something that's deforming and moving through a fluid. Maybe we just want a complicated biological structure that is relatively rigid, doesn't move, and flow moves past it. So there are a number of steps for doing something like this. And what I'm thinking is something like, let's say we want to do a numerical experiment where we have something like a wind tunnel or a flow tank, and we put our structure in it. Maybe it's a coral, for example, or a crab shell, horseshoe crab, like I showed earlier. And then we want to numerically simulate the flow around it or through it. So the steps that are going to be involved in this are object creation. So how do we get a 3D object? We can look at 3D repositories, and there are quite a few, so I'll show you some in the next slides. We could do photogrammetry, and let's say that we're looking at a cactus, right? So we could go out and take 100 pictures of the cactus from different angles, and then use photogrammetry software like Autodesk to reconstruct it as a 3D object. We could do 3D scanning. Maybe we have a 3D scanner. I've heard that the iPhone apps for 3D scanning work amazingly well now. So those are some options. We could also design our object from scratch using CAD. So something like Autodesk Fusion, SolidWorks, FreeCAD, or Blender. The Autodesk products are free for academics. SolidWorks, you need your university to have a license. And then there are some free open source like FreeCAD and Blender as well. Now, in particular, let's say that you go somewhere and you grab a 3D STL file of a coral that someone made using photogrammetry or 3D scanning. You're probably going to have to repair and smooth it to actually do a simulation. And so a really nice software product is Mesh Mixer. And I'm going to show you Mesh Mixer. Autodesk Fusion 360 can do this, and I'm sure that there are other products as well, but these are the ones that I've used. All right, now that you have your nice repaired and smoothed object, you have to mesh it. And so Gmesh is free, and that was used in the reproducibility paper. There's also Cubic Chloroform, which is now free to academics. And if you're using a bundled commercial software like COMSOL or ANSYS, they will have their own mesher. Now, let's say you do your simulation using OpenFOAM or COMSOL or ANSYS. Well, you can visualize it in the package if you're doing COMSOL or ANSYS, or you can use some of these visualization softwares like this, which is free, ParaView, which is free, or TechPlot, which costs money and only runs on Windows, but is very nice. Okay, so just quickly, don't have a ton of time left, but let's say that we want to do a simulation in COMSOL, flow past the coral. There are tons of 3D file repositories like Thingiverse, Imagine, Libre 3D. They aren't, of course, scientific, but if you're just playing around these, it's nice to grab different things from these repositories. There are also numerous scientific repositories. So NIH has one that has a lot of vessels, arteries, valves, the heart, and so forth. So if you are interested in medical applications or you have students, undergrad researchers who are, this is a very nice resource. There's also digital morphology, Morphosource, Phenome 10K, GB3D, Smithsonian 3D, University of Michigan online repository of fossils. So more and more you can find these 3D repositories and get lost in sort of the vast amount of data that's there, which is pretty cool. So I'm going to switch back to my other computer really quickly and just wrap this up. So I can't remember where I found this coral. I think it might have been Thingiverse because it was a few years ago. But I just want to show you really quickly. Here is the coral. If I hit W, so this is open in Mesh Mixer, I can see the mesh. It's really quite fine. It's too fine to easily simulate. Also, ideally, all of your elements are going to be, well, they should look like equilateral triangles. And so there are some issues with these really acute triangles. So I will probably want to remesh this and smooth it. Mesh Mixer has really nice tools for doing this, which is maybe its own, it's definitely its own tutorial. All right, so here I've selected the whole coral and I can smooth it. I think my computer is unhappy. All right, so I've smoothed it quite a bit. I could smooth it less. Well, and at some point you just end up deforming it beyond recognition. So it's an art, like how much can you smooth it and have it still somewhat resemble reality and be something you can simulate. But more details on that for another day. When I was doing this originally, here is my smooth coral, which is quite a bit smoother, but I always suggest starting with something easy. So something extremely smooth and then making it more realistic so you can sort of wrap your head around how difficult this process is going to be. Now here is COMSOL. This is what the interface looks like. And the nice thing about COMSOL and ANSYS is that it really walks you through this whole process of doing a simulation and everything is packaged together. So it has its own geometry creation. Here you might be able to see I've imported this coral and then I put a box around it. And so it's like a coral sitting in the bottom of a flow tank. The slides that I share sort of step through more of each of the steps than I have time to do now, but you basically import your coral, you put a box around it, you might need to scale and move stuff. And then what you're simulating is the fluid. So you want a box and you want to subtract the coral out of it, right? So then you have this domain of water, and how it knows that the coral is there is because there's, essentially a boundary, and you're not simulating the fluid in the region of the coral. It has a lot of materials that are loaded, so you can pick air, water, mineral oil, other such things. You can pick your fluid solver. So I'm doing a large eddy simulation since the Reynolds number here is, I think it's around 10,000. So it's a bit turbulent. There's a creeping flow if we were at extremely low Reynolds numbers. But again, you have to know your Reynolds number and what solver you want to use. You can pick all of your boundary conditions. So you have an inlet or an outlet. You can mesh it. So here's our mesh. This is a pretty coarse mesh because I wanted to get it to work. And then you run your simulation and you have results, which I've already done. So here, okay, the flow is moving left to right. Here's our coral sitting here. We have this wake that's developed. Okay, so the nice thing about ANSYS and COMSOL is that you have this lovely graphical or user interface, and everything you need is there. You can create your geometries. You can import different fluids. There are a number of different solvers. You can mesh it. You can solve it. You can visualize it. Open source software, opt-in. All right. It's not all packaged together. You don't have a GUI, and you're using different software for each piece of the puzzle. OpenFOAM is somewhere in between because there is meshers and so forth that are available within it, but yeah. So I think I will end there, and I'm happy to take any questions. So very quick intro to CFD.
That mesh size is something that you have got to set early on, or is it like a process of refinement as you figure out the stuff that you don't see or you don't see you need?
Yeah, so I mean, the way that I have done this is because there are so many things that can go wrong. You start with a really coarse mesh because it's really easy to choose a mesh that's so fine that your computer will just freeze. It requires too much memory, or it will take 10 hours to do the simulation and then you realize that you've done something wrong. Or that, if you import something like the coral, and you've got to do this Boolean subtraction, that will fail because the mesh of the coral is too refined. So I recommend just start with a super coarse mesh, smooth everything a lot, get it to work, and then go back through and slowly add more refinement so you can get a handle on what you can get away with. And you'd be surprised just how difficult it is. If you're looking at really complex biological structures like sea fans or things that have bristles, being able to mesh and simulate all that complexity is tough.
Yeah, there's a recent paper, they did some ultra fine mesh of flow through a glass sponge. I think some folks at NYU.
Yeah. So that was very nice, but they did not take a 3D scan of a glass sponge. They created their own geometry, which made it nicer. And they used the fact that the structure is somewhat periodic. And it was still very hard. Like, that was an amazing paper, but they had to make a lot of assumptions to get that to work by just focusing on like one segment, assuming it was periodic, and then making it a bit nicer by generating the structure with CAD versus a 3D scan.
So it wouldn't have worked if they used the actual museum specimen, you think?
If you zoom in really close, you can get it to work. I don't know that I'll say I couldn't get it to work. Maybe they could get it to work. But I could not get it. I would not be able to get it to work with a museum specimen. Yeah.
Laura, we have one question in the Q&A box. Are you able to see it?
Oh, let me open it.
With CFD, can you take us back to box? What makes these wrong models useful, where useful is a continuous metric? Thanks. For
Yeah.
Yeah.
Oh, yeah. So I think just like with physical models, which folks might have a little more intuition for, or even just anything experimentally that's in the lab instead of the field, you don't have all that complexity, but you can really sort of narrow down on what matters. And if you're just changing one thing at a time and not considering things like the fact that, let's think of the glass sponge example, right? So in the ocean, there are currents, the flow is unsteady. Let's say you take that away, which makes it easier to understand. And then you can ask some things like, all right, we have this very complex structure, and if we remove some of it, so the glass sponge has these helically wound ridges, what if we took those away, right? And then add them back in. How does that change the flow patterns? How does that change the drag? And one could always say like, well, you're looking at this and it's not like a turbulent, unsteady flow that a glass sponge would really feel. But if you add all that complexity to it, it's just hard to even tease out what's happening. So it's like a starting point, right? Like in this controlled setting, we've set up our model in these ways, we add these things and then take them away. And we can see how that changes what's going on and start to get some intuition for how each of the pieces matter in isolation. And then again, like ideally someone would go back later and check like, okay, if you do have unsteady currents and turbulence, do the helices still play the same role or not? Right. I hope that answered the question. I think another good example of that too is with insect flight. There were people who did simulations of just a fixed wing in 2D. And you can show that this doesn't generate enough lift and that you end up with stall when you have a high angle of attack. And so that, in a way, is not a good model. But in another way, it was extremely informative because it showed you that, okay, the 3D rotational motion and delayed stall are really important for insect flight.
I have a general question. This way outside my field, so apologies if it's somewhat superficial question, but how much more complex does it make these if you are dealing with a compliant object? So where the fluid actually deforms the object you're studying.
Yeah, I mean, so you can definitely do that using the immerse boundary method. And the nice thing about, let's see if I can share and just show something really fast. All right, so one of the really brilliant aspects of the immerse boundary method is that, let's say you're solving for the position and the elastic force on each of these nodes, and this is a moving, deforming, curvilinear mesh. It does not have to match up with the fluid domain. So the immerse boundary method provides a way of sort of transmitting the forces from this discretization, this curvilinear mesh, to this fixed Cartesian grid. The challenge when you do that, though, is that you have to take a pretty small time step. So if you're simulating something over a second, maybe you have to update your solution every 10⁻⁵ seconds. So that really slows things down. And so if you weren't doing it this way and you were using COMSOL, you would probably get your solution faster. You would take bigger time steps. But let's see. If you're using something, if you're not doing an immerse boundary implementation in one of the commercial software, then you have this fixed cred. And so if you move or deform your object, you might have to remesh. And so when you are trying to mesh a complicated structure like this coral, you realize how long that takes and how hard it is and how often it fails. So then doing that, every 10th of a second is not going to work so well. So I guess the quick answer is you can do it, but there are costs and there are different ways that you can do it. Overall, it's going to slow things down, but it's feasible.
Are some of the software frameworks better at handling that kind of simulation where you have compliant objects?
Yeah, so I mean, I think all the immerse boundary ones that so the IBAMR, IB2D, but so there's another interesting thing as well. There are a lot of people who have really good software for doing this that's in-house and may or may not be available for other people to use. And so then it's, okay, why is it not available? And that's complicated too. So there aren't really good funding mechanisms to pay people to develop their own software to make it user-friendly for other people. And that's a huge challenge. So a lot of the people who are doing this, making their code available, are doing it for free with the hopes that they'll get enough collaborations down the road to make it worthwhile. But NSF programs that fund such work, I think, are hard to find. And so that's my impression anyway. Someone could tell me that they'll fund such work and that would be great. So then, you could definitely find beautiful publications and simulations with deforming objects, and they have in-house code, and maybe they'll share it and maybe you can figure it out and use it. It's hard to say. I know that OpenFOAM now has immerse boundary modules. I have not used them myself, so I'm not sure how easy or hard they are to use. ANSYS has ways of doing fluid structure interaction, but I also have not used them yet. So I think in general, I would suggest not starting there, just so you can learn things and figure them out and then adding that complexity.
That makes a lot of sense. Thanks. All right. It is past 9, so unless we have other questions, maybe time to wrap up.
All right, thanks everybody. I gotta go teach.
Thanks so much. I really enjoyed those presentations, and it'll be, I think, a great resource for the community to have these videos online for graduate students who are starting out in the field and things like that. So thank you very much.
Yeah, thanks for inviting us, and I'm happy to share. I had so many links on my slides. I will share them because I know I kind of bombarded everyone with it.
That would be great if you could share the slides, and we'll include that on the SIMS website with this session as well. Thank you so much.
Excellent. Great. Well, thanks everyone.